from the GDCB department right here at Iowa State University. She is the Walter E. and Helen Park Loomis Professor of Plant Physiology, the first one of those at Iowa State. And she received her BSc with honors in biochemistry from the University of Birmingham, England, and her PhD in biological sciences from University of Warwick. Then she postdoced at the MSU DOE Plant Research Laboratory at Michigan State. And she came here in 2001 and has done great research. That's why she's got this named professorship. And she works on autophagy, and I'm interested in her work because that's regulated by a complex called TOR, which controls translation. And since I work on translation mechanisms, it sort of ties it in. And also, this stuff, autophagy, can relate to plant pathology because some plant viruses take it over, utilize it to get rid of host proteins they don't like. But we won't be hearing about that today.
So thanks for inviting me to tell you about our research. And hopefully, even though I'm not a plant pathologist, you'll see that there are some connections, as Alan mentioned, to plant pathology and plant pathogen interactions, even though in my lab we mostly focus on abiotic stresses. And so I'm going to talk to you about this process called autophagy, also called autophagy, depending on who you talk to. And there have been many discussions in the field about the pronunciation, so I think either is correct. But autophagy literally means self-eating. So auto means self, phagy means eating. So this is a process that happens within a single cell, within a plant or an animal or basically any eukaryotes, where proteins and organelles and other cell constituents are degraded. In plants, the basic process, when it's activated, involves some portion of the cytoplasm. And this can be organelles like mitochondria, large structures, things like ribosomes or proteasomes, or individual proteins. And these are surrounded by a membrane structure. And this membrane forms a cup shape around the cargo, the material that's going to be degraded, and that cup elongates and eventually fuses to form a complete vesicle. And that's what we know as the autophagosome, this structure here. Notice that it has a double membrane, which is really unusual for a vesicle that transports materials. This vesicle, the autophagosome, then moves to the vacuole. The outer membrane of the autophagosome fuses with the vacuolar membrane, and the inner membrane and its contents end up inside the vacuole. Now remember, the vacuole is a place where many different hydrolytic enzymes are found. And so once material gets inside the vacuole, it's degraded very rapidly back down to its building blocks, things like amino acids. These are exported from the vacuole, and they can be reused by the cell. It can be selective or non-selective, and what we mean by that is that either bulk cytoplasm, pretty much anything, can be degraded in some circumstances. In other circumstances, very specific cargo, an individual protein or an individual organelle, can be selective for degradation. Okay, so degradation pathway, question is, why do we care? Why is this important? Turns out that autophagy functions in a lot of different processes in plants. It occurs in response to many different environmental stresses. And this is really where my lab focuses. We look at abiotic stress, environmental stresses, things like nutrient deficiency. Turns out that during nutrient deficiency, autophagy is activated, breaks down cell components to provide nutrients to the cell. So the cell, in this situation, literally is eating itself. It can also provide, those breakdown products can also provide TCA cycle substrates to provide energy for the cell, because in a low nutrient situation, it's likely that ATP levels are dropping as well. It's also activated under actually a wide range of other environmental stresses, from salt stress to oxidative stress to heat stress, many different situations. And we think that in a lot of these cases it's required for damage clearance. These stresses cause damage to macromolecules, including proteins and lipids. And these can become toxic to the cell if they accumulate. And so autophagy can degrade these molecules as a way of clearing damage and allowing the cell to survive. It's also activated upon pathogen infection. And it's been maybe not controversial, but there have been a lot of different results that have been presented as to what the function of autophagy might be upon pathogen infection. There have been some labs that have shown that it's required to restrict programmed cell death to the site of bacterial infection, for example. And other labs have shown that it's required for bacterial infection. In the case of viruses, autophagy in some cases is required to help the plant to survive upon viral infection, which actually enables more viral replication. And in other cases, it's been shown to degrade viral proteins and prevent viral replication. So depending on the pathogen and even the age of the plant and the type of plant that you look at, autophagy actually can have different roles. And so because of this complexity, we really don't study pathogens. We leave that to people that know what they're doing. We stick to the abiotic stresses, where it's still complicated, but in most cases, at least we can say autophagy is a cell survival process. It helps the cell to survive during these stresses. It is also sometimes developmentally programmed. This is most obvious during senescence. So autophagy is strongly activated during senescence in leaves, where it breaks down leaf proteins and organelles, probably to remobilize the nutrients to developing seeds, for example. And it's also been implicated in various forms of programmed cell death. And most of what I'm going to show you is molecular and cellular phenotype data. So I just thought I'd point out to you that you can actually see the effect of autophagy at the whole plant level. So you can see whole plant phenotypes when you disrupt the autophagy pathway. So it is significant for plant survival and not just significant on the individual cell level. And so in this picture, these are Arabidopsis plants. We mostly work with Arabidopsis, and then we have some work in maize. So these are wild type Arabidopsis plants on the left. RNAi, these are plants where the autophagy pathway has been disrupted by RNA interference to knock down, almost completely knock out expression of a gene that's required for autophagy. And so basically these plants on the right have no autophagy pathway. So under normal conditions, they look almost the same as wild type. There's really not very much difference. But when we start to stress them, we can really see why autophagy and when autophagy is important. So upon salt stress, wild type plants are surviving pretty well. You can see our autophagy defective plants are pretty small and grow much less well than the wild type. So it's important for tolerance of salt. And at the bottom, you can see plants exposed to drought stress. We basically just stopped watering the plants. And wild type are growing and surviving pretty well. And at this point, our RNAi plants are almost dead. So these plants at this point, the RNAi plants that don't have autophagy are not going to recover if you rewater them. So we see fairly dramatic effects on stress tolerance if we affect the autophagy pathway. So this tells us we are really looking at a process that's actually important to the plant, even though we're mostly looking at cellular phenotypes. And so over the years, some of the questions that my lab has addressed are things like, simple questions like, under what conditions is autophagy activated? And the reason that this was an important question initially is because autophagy historically has been considered to be a response to nutrient deficiency. For many years, that is how it was studied, and that is where all the original basic work was done. And so we wanted to define things that required autophagy for plant tolerance. And so that includes those that I showed you on the previous slide. We wanted to know what the functions and the substrates of autophagy under these conditions are. So we came back to the nutrient recycling and damage clearance aspects, again, that I showed you on the last slide. And then what I'm going to talk about today is a project that we've been working on for several years now, thinking about how autophagy is regulated. So the idea is that autophagy is activated and important under this wide range of different conditions. So how is it activated? What regulates the activity of this pathway? We know that under normal conditions, it's active at a very low level, and it's very strongly upregulated during stress. So we wanted to find some of the signaling components that trigger this activation of autophagy under these different conditions, and to also figure out, is it the same components under all these stress conditions, or does each stress have a different type of signaling pathway to activate it? And you'll see it's kind of a combination of those two. And so I wanted to start out with a protein complex that Alan actually mentioned in the introduction, because he has some interests in this as well. And that is a protein complex called the targeted rapamycin complex. This is conserved, it's found in animals, it's found in yeast, it's found in plants, throughout eukaryotes. And it consists basically of 3 subunits. What I'm showing you here is the structure of the complex from animal cells. We don't have a structure of the plant complex. But one of my students did, actually Jen, she's sitting there in the back, she took the animal complex and she was able to model the plant TOR complex onto it. So it looks like structurally these are conserved as well as just sequence. And so the 3 components are the 3 proteins that make up the TOR complex are TOR itself. This is the catalytic subunit. TOR is a protein kinase, so it phosphorylates other proteins and changes their activity. And that's really the function that we'll be looking at. And then 2 accessory subunits, LST8 and Raptor. And I'll also bring back Raptor. This is a kind of a regulatory subunit of the kinase, so it controls the protein activity. And the reason that we were interested in this complex is that it controls cell responses to nutrient concentrations. And we knew that this was a major regulator of autophagy. So nutrients, high nutrient concentrations, activate the TOR complex and the TOR kinase activity. This controls growth, it activates growth, which means that when nutrient concentrations are high, the cells or the organism can grow. And this growth includes things like translation, which Alan's interested in. And it blocks catabolic pathways like autophagy. So this makes sense. And this was known in animal cells before we started looking at this in plants. Upon nutrient deficiency, so in the case of a plant, this would be something like nitrogen deficiency, macronutrient deficiency. Nutrient deficiency blocks the activity of this complex. This means that growth will be inhibited and autophagy will be activated. And so, first of all, we wanted to confirm that autophagy really is activated by blocking TOR in plants. But the more interesting question to us, assuming it was, is what about other stresses? TOR responds to nutrient concentrations. Autophagy is activated with a wide range of stresses. So do these other stresses activate autophagy by a completely different mechanism, independent of TOR? Or do they work also through TOR by inhibiting TOR activity and then allowing activation of autophagy? So we first confirmed that TOR really does regulate autophagy in Arabidopsis. So we're using Arabidopsis as our model plant. And here I want to show you some of the assays we can use to measure autophagy activity. One of the major ways is using fluorescence microscopy. And so if you remember, the autophagy pathway involves formation of these vesicles that transport material to the vacuole for degradation. These vesicles are autophagosomes. And so we can label the autophagosomes and count the number. And so the number of autophagosomes we see in a cell gives us a measure of activity of the autophagy pathway. And we have 2 different ways of labeling these autophagosomes. One is using a fluorescent dye called MDC, monodensyl cadaverine, and this accumulates specifically in autophagosomes. Another is fluorescent protein fusion. So a fusion of green fluorescent protein to a protein that localizes to the membrane of autophagosomes called ATG8. And so when we express this fusion in plants, we can also see fluorescent autophagosomes when autophagy is activated. So we can use those two assays basically interchangeably. The disadvantage, of course, of the GFP ATG8 fusion is that we have to make transgenic plants to express a marker. And so here, we've taken wild-type plants, and we've taken mutants, Raptor 1A and Raptor 1B. Now, Raptor, you remember, is one of the components of the TOR complex. And so we're basically shutting down TOR activity. We can't knock out the catalytic subunit itself, the TOR subunit, because that's lethal to the plant. So presumably in these Raptor mutants, we have some residual TOR activity that allows survival. And what we see, TOR, of course, we hypothesize it negatively regulates autophagy, positively regulates growth. And you can see on the fluorescent microscopy, these little dots that the arrows are pointing to down here in this Raptor mutant. These are autophagosomes. And these are under normal conditions. So in wild-type plants, we see very little in the way of autophagosome formation. Actually, the Raptor 1A, there are two genes in Arabidopsis for Raptor. The Raptor 1A knockout has very little effect, but if we knock out Raptor 1B, we see this pretty strong activation of autophagy. And we can quantify that by counting the autophagosomes and then drawing a number of autophagosomes per frame, by which we mean per image that we take. Very few in wild type. Raptor 1B has many autophagosomes. So we can see that if we disrupt Raptor, then we have activation of autophagy. We can also use AZD, which is a TOR inhibitor, directly added to the plants, and we basically see the same thing. We see a large number of autophagosomes accumulating, telling us that TOR is a negative regulator. So then we went on to look at other stress conditions. What happens if we change activity of TOR during stress? To look at this, we overexpressed TOR in transgenic plants with the idea that increased TOR activity would repress autophagy under stresses where TOR was required for activation of autophagy. And so we did the same kind of assay where I have now wild-type plants along the left, overexpression of TOR transgenic lines on the right. And hopefully you can already see from these images that upon sucrose starvation, we're activating autophagy. Each of these little bright dots is an autophagosome in wild-type plants. In our TOR overexpression lines, we see very few autophagosomes, suggesting that overexpression of TOR is repressing autophagy under these conditions. And we can quantify this again by counting the autophagosomes. And first of all, looking under nutrient starvation, conditions in which we would expect TOR to be important for autophagy, we do see in wild-type plants, these red and the green bars show activation of autophagy under sucrose and nitrogen deficiency conditions. All of our different TOR overexpression lines, I'm showing 5 different lines here, these all have repression of autophagy under these nutrient deficient conditions. So it looks as though, as we'd expect, TOR negatively regulates autophagy during nutrient deficiency. What we didn't necessarily expect as much is that when we looked under salt stress or osmotic stress, we saw the same thing. We saw again, this activates autophagy in wild type compared with the control in blue, but in all of our TOR overexpression lines, we see no activation of autophagy at all under these conditions. So this tells us that TOR is required, repression of TOR activity is required for activation of autophagy under salt stress. But this doesn't happen under all stress conditions. We then looked at other stresses that we know activate autophagy, oxidative stress and ER stress. And what we see is that under these conditions, even in our TOR overexpression lines, autophagy is activated just fine, as you can see with these red bars. Same number of autophagosomes basically in the overexpression lines as in wild type. That's for oxidative stress. And we see the same thing for ER stress. And I'm going to come back to the ER stress idea later. So to put this together, what we found is that TOR is a negative regulator of autophagy, and we also know from other work that it activates growth in plants, and that nutrient stress and salt and osmotic stress all repress TOR activity, and that this leads to activation of autophagy in these stress conditions. But we've also seen that there are other stresses, oxidative stress and ER stress, that also activate autophagy, but independently of TOR. So we have at least two pathways for activation of autophagy. Now, notice that I've added auxin, the plant hormone, onto this slide. Auxin has been shown to inhibit, well, okay, so auxin affects TOR activity, but this has never been linked to autophagy previously. So auxin can activate TOR, and this will lead to changes in translation and growth. So we wanted to know whether auxin could also affect autophagy activity through TOR. And we saw basically exactly what we would expect if auxin was just acting as a regulator of TOR. And so here we're using this GFP ATG8 fluorescent protein fusion as a marker for autophagosomes, and we see very strong activation of autophagy. All these little bright dots are autophagosomes. We see very strong activation under all of these stress conditions. If we add auxin, NAA, we pretty much completely block the activation of autophagy. So presumably, the effect of auxin upstream of TOR is leading to a downstream consequence on autophagy. And when we look in the conditions where autophagy is activated independently of TOR, now we see that auxin has no effect. We can still get activation of autophagy. And to kind of confirm that this happens through the TOR signaling pathway, we used this inhibitor of TOR, AZD, and found that auxin has no effect on activation of autophagy by this inhibitor, which tells us that auxin is upstream of TOR in this pathway. And we did a similar experiment with a raptor mutant that also kind of has constitutive autophagy even in the absence of stress. So this tells us that auxin activates TOR, and this leads to repression of autophagy. So we now have a hormone in our signaling pathway for activation of autophagy. We don't really know where it fits in with these other stresses, and that will be interesting to look at in the future. Another component that we were interested in studying to figure out if it regulates autophagy is the SNRK1 complex. This is another protein kinase complex. And the reason that we were interested in is this is another sensor and regulator of nutrient and energy responses in plants. And so it controls responses to energy depletion. And so it's a kinase complex, and the catalytic subunits, it has two possible catalytic subunits, kin10 and kin11. And we just asked simple questions, does SNRK1 regulate autophagy? And if so, what's its relationship to TOR and the regulation of autophagy by TOR? And we found that yes, SNRK1 does affect autophagy activity. So here we're using overexpression lines that have increased SNRK1 activity. We're overexpressing the kin10 catalytic subunit of this enzyme. And again, we're counting autophagosomes, and we find that even in the absence of stress, we have activation of autophagy in these overexpression lines, whereas in wild-type plants, we see very little autophagy because there's no stress. And we can do it in two different ways. We can stain roots of plants, the wild-type or the overexpression lines, with the MDC dye. We can also make protoplasts from leaves of these plants and express ATG8 fused with GFP as our autophagosome marker in a transient expression assay. So in both of these assays, the MDC staining of roots and the transient expression of our fluorescent marker in protoplasts from leaves, we see the same thing. We can also use a chemical that activates the SNRK1 complex, and that also has the same effect. It activates autophagy. So we think we're pretty confident that SNRK1 activates autophagy in Arabidopsis. We can also do the opposite experiment and make a mutant where we've lost content, the catalytic, so we've lost SNRK1 activity. In these mutants, what we see is that basically these mutants are unable to activate autophagy under almost all of the conditions that we've tested. So we tested salt stress here, and I'll just walk you through. So in salt stress, in wild-type plants, salt stress activates autophagy compared with the control. In the Kin10 mutant, we see no activation of autophagy by salt stress. And we see the same pattern in sucrose deficiency, oxidative stress. Mannitol, you can get a little bit of activation of autophagy. We're not sure why mannitol or osmotic stress acts a little bit different. Nitrogen deficiency, you see a blockage of autophagy, and this is an ER stress inducer. So based on this, we can see that pretty much every stress we look at, this Kin10 mutant is blocked in activation of autophagy. And so we wanted to look at the relationship with TOR. Remember, TOR is a negative regulator of autophagy. SNRK1 seems to be a positive regulator of autophagy. So how do these fit together in a pathway? And so the first thing we did was block TOR activity using this inhibitor AZD in a Kin10 mutant. And so I think it's easiest to look at the graph. In wild-type plants, AZD, you add it, activates autophagy because TOR activity is blocked. In the Kin10 mutant, normally Kin10 is blocked in activation of autophagy, but our TOR inhibitor can still activate autophagy even in this Kin10 mutant. So that suggests that TOR is downstream of Kin10 or of SNRK1 in this pathway. We did the opposite experiment where we blocked SNRK1 activity and we used an inhibitor, drahila 6-phosphate, that inhibits SNRK1. And we did this in wild-type plants, and we did it in a raptor mutant. So the raptor mutant, even under normal conditions, usually has high levels of autophagy. The SNRK1 inhibition had no effect on this. So this again suggests to us that SNRK1 is upstream of TOR. We went on to do a bunch more experiments, the same kind of combinations where we would activate one pathway and inhibit the other to try and confirm the order within this pathway. So here we've overexpressed TOR, and we found that this activator of SNRK1 has no effect. We've added auxin to Kin10 overexpression lines. We've added ACAR, which is the SNRK1 activator, in combination with auxin. And basically, everything that we do suggests to us that TOR is downstream of SNRK1 in a signaling pathway that leads to autophagy. So, just to show you how this works, so we have SNRK1, and this would inhibit TOR, and then TOR inhibits autophagy, so SNRK1 would activate autophagy by inhibiting the activity of the TOR complex. Now, I haven't put these stresses in upstream of SNRK1 because we're still not really sure how this all fits together. One thing that we do know that was obvious to us, though, is that SNRK1 also activates autophagy independently of TOR, because SNRK1 is involved in activation of autophagy in some stress conditions. These TOR-independent stresses, oxidative and ER stress, still require SNRK1 for activation. So this was something that was kind of a surprise and interesting to us to try and understand how these stresses that are TOR-independent could activate autophagy. And so we actually went on to look at these stresses in more detail, and particularly ER stress. And for those of you that are not familiar with ER stress, it's a condition where unfolded proteins accumulate within the endoplasmic reticulum. And this is because proteins can't fold fast enough. There isn't enough folding machinery there. And so if proteins are being misfolded or unfolded by stress conditions too quickly, the folding machinery can't keep up, and you end up with this accumulation of unfolded proteins. And this causes stress on the endoplasmic reticulum and in turn on the whole plant. And it's caused by, traditionally it's been studied as an effect of heat stress. This is probably the most common way, and this is really how we've looked at it. So heat stress triggers protein unfolding and it triggers ER stress. But it's also caused by salt stress, pathogen infection. Viral infection is known to cause ER stress in some circumstances. And in the lab, we can use chemicals. We can use DTT or tunicamycin. And these are chemicals that basically prevent protein folding within the ER. And so they artificially trigger ER stress. And it's easier to control these to use them in the lab. And so just to show you, this is ER stress. We're using these two chemicals, DTT and tunicamycin and TM. And again, same assay to look at autophagosomes. We see that this ER stress causes accumulation of autophagosomes, and so ER stress triggers autophagy. And we've done some work looking at the function of autophagy during ER stress. And what we found is that the ER is actually delivered to the vacuole for degradation during ER stress. So here we've used GFP. So the green fluorescence here is GFP fused to an ER localization signal. So basically the green fluorescence is labeling the endoplasmic reticulum. And then we've now fused our autophagosome marker with cerulean, which is a different color fluorescent protein, so we can do double labeling. And we've seen that if you look at the lower panel here, we've blocked degradation in the vacuole using a chemical so that we can see the autophagosomes more easily. And we see that the GFP HDEL, the ER marker, accumulates inside the vacuole in little structures, which when we look at our autophagosome marker, turns out are autophagosomes. And so what we conclude is that the endoplasmic reticulum is being delivered to the vacuole and degraded by autophagy during ER stress. And we've been able to show that this is specific to ER stress, as this doesn't happen in nutrient deficiency conditions, for example, even though autophagy is activated under those conditions. And so we were interested in what triggers autophagy during these ER stress conditions. We know that SNRP1 is involved. We know that TOR is not involved. We know a little bit about ER stress signaling in Arabidopsis. And this is from work, a lot of it is from work done by Steve Howell in GDCB. And we've collaborated on this part of the project with Steve and with his lab. And the ER stress signaling pathway, which is known as the unfolded protein response, starts when unfolded proteins within the ER are recognized. And in plants, there are two separate pathways for activation of this unfolded protein response. One is a membrane-bound transcription factor called BZIP28. And upon ER stress, this is cleaved by a protease that releases it from the membrane. It then can enter the nucleus, where it acts as a transcription factor to upregulate things like molecular chaperones that help in protein folding and help to counteract the ER stress. A second transcription factor called BZIP60, which has similar kind of overlapping functions in upregulation of chaperones and other ER stress factors. This is also bound to the ER membrane, but instead of being proteolytically cleaved, the mRNA encoding this transcription factor is spliced only upon ER stress by a membrane-bound splicing factor called IRE1. So when unfolded proteins accumulate, the cells recognize ER stress. IRE1 splices the messenger RNA for BZIP60. That produces a transcription factor which is no longer bound to the ER membrane, which enters the nucleus and activates transcription. Okay, so none of this seems to have anything to do with autophagy. But we knew that these factors were all involved in ER stress responses, so we hypothesized that maybe some of them would be required for activation of autophagy. So we first very simply looked to see whether unfolded proteins were important. And to do this, we used two approaches. One was to try and decrease the amount of unfolded proteins upon ER stress. And to do this, we use chemical chaperones. One example is this chemical chaperone called PBA. And we found that if you look at number of autophagosomes, here's a control, very low. Pudicomycin is one of these chemicals that activates autophagy because it causes ER stress. If we add pudicomycin and we add this chemical chaperone, we're back down to background levels of autophagosomes. So this chemical chaperone blocks the increase in autophagy during ER stress. Same using a different chemical to trigger ER stress, ATT. When we look during sucrose starvation, PBA has no effect. So it only affects autophagy that involves unfolded proteins like these ER stress responses. We did the same experiment by overexpressing BIP, a molecular chaperone, and we saw the same result. The second approach was to overexpress a mimic of unfolded proteins in the endoplasmic reticulum and see whether that could trigger autophagy. So we use this protein called zeolin, which is unable to properly fold. It gets stuck in the endoplasmic reticulum and triggers an ER stress response. And when we express this protein in the absence of any other ER stress trigger, we found that indeed we got activation of autophagy just by the expression of an unfolded protein mimic. So we concluded that, that unfolded proteins are important in activation of autophagy. So then we went on to look at some of these downstream factors that signal in response to ER stress. I'm not going to show you the data. We looked at BZIP28. This doesn't seem to have any effect or any role in autophagy whatsoever. We couldn't see any effect of mutants. But we saw, when we looked at IRE1, we saw a different story. We found that IRE1 is actually required for activation of autophagy during ER stress. There are two IRE1 genes in Arabidopsis again. It turns out that IRE1B is the one that's important for autophagy. We don't know whether this is really a difference in function or just that this is the most highly expressed gene. Not really sure. But what we see is that in the white bars are activation of autophagy upon ER stress. These gray bars show you what happens in IRE1B mutant. So autophagy is pretty much completely blocked when we activate using ER stress agents. Again, sucrose starvation activates autophagy just fine in this IRE1B mutant. If we look at an IRE1AB double mutant where both genes are knocked out, we see the same thing as in an IRE1B mutant. And so we wanted to understand what it is about IRE1B that's important for activation of autophagy. So I mentioned it's actually a splicing factor, a non-conventional splicing factor, because it acts at the ER membrane and not in the nucleus. And it very specifically splices just this single messenger RNA encoding the transcription factor that then responds to ER stress. If we look at the structure of IRE1B, it has a luminal domain, which is in the ER lumen. This is the region that senses the unfolded proteins and recognizes the ER stress condition. The cytoplasmic domain actually has two different activities. It has kinase activity, so this is a protein kinase. It also has ribonuclease activity. And the ribonuclease activity, of course, is required for the splicing, this messenger RNA splicing. And so we wanted to tease apart, specifically, can degrade messenger RNAs that are present at the endoplasmic reticulum. And presumably, this decreases the amount of protein being translated at the ER and will decrease the unfolded protein load. So we also wanted to distinguish, and that's called RID, which is so this more general RNA degradation pathway. We also wanted to distinguish between these two pathways. So we started out looking at these point mutations, and basically the conclusion we came to was that the ribonuclease activity was important for autophagy, but the kinase activity of IRE1 is not. And that's because if we look down at the graph here, so in wild type we get activation of autophagy by ER stress. In an IRE1 mutant, we get no activation. We can express back the wild type IRE1, and get activation again. But if we express back the two mutants that block the ribonuclease activity, we no longer have activation of autophagy. If we put back the kinase dead mutant, we get autophagy just fine. So this tells us that the kinase activity actually isn't important. And our original hypothesis was that maybe it was this kinase activity that was activating autophagy, because we know that autophagy is activated by phosphorylation. Turns out we were completely wrong, and it's the ribonuclease activity that's important. And so we did this. So this was in transient assays in leaf protoplasts. We also did it in stably transformed plants, where we'd taken mutant plants and stably expressed these mutants. Basically, we see the same thing. We see that the kinase mutant can activate autophagy, the two ribonuclease mutants cannot. And so basically, when we look back at our scheme, we've been able to rule out this branch where protein phosphorylation by the kinase domain activates autophagy, and we're left with the ribonuclease activity, which again has two possible features. So it has splicing of BZIP60 mRNA and this more general RNA degradation pathway. So to distinguish between these two, we looked at BZIP60 mutants. If it's BZIP60 splicing that's important, then autophagy should be blocked in a BZIP60 mutant, but we found that 3 different independent knockout mutants of BZIP60 could all activate autophagy in response to ER stress with no problem. Again, something of a surprise to us. So we could also rule out BZIP60 splicing activity of IRE1. That leaves this more general mRNA degradation pathway as being important for activation of autophagy. So we wanted to identify, are there specific messenger RNAs that have to be degraded to get activation of autophagy? Or is it just general messenger RNA degradation that's important? And so to address this, we took IRE1 mutants compared with wild-type plants, and we did RNA-seq to look at gene expression. And we wanted to find messenger RNAs that were degraded in wild-type plants, but were not degraded in IRE1 plants. And these would be candidates for being rid substrates that are degraded by IRE1. And so we did this, and we just got a list of genes, and we picked the top 12, because that was how many we could handle. And we overexpressed each of these top 12 genes in Arabidopsis protoplasts and looked at their effect on autophagy. The idea is, if these messenger RNAs have to be degraded in order to activate autophagy, if you overexpress them, it should block autophagy. And so we looked at all 12. And what we found was that most of them had no effect. But if you look at these 3 in the middle, you can see that overexpression, and so we're looking at tunicamycin in the red bars, so this is under ER stress conditions, you no longer get activation of autophagy upon overexpression of each of these 3 messenger RNAs in the protoblasts. And these 3 genes were then our candidates for having messenger RNAs that had to be degraded to activate autophagy. And we looked at what the 3 genes encoded. One was a beta-glucosidase 21. This is a protein that's found in ER bodies. And the other 2 were lipid-related proteins. None of these have a really well-defined function that we could then test to see whether that function was important in regulating autophagy. But they do all seem to be involved somehow in lipid and membrane dynamics and remodeling at the endoplasmic reticulum. So it gives us some hope that they're involved in autophagy. And we wanted to know whether it was just the messenger RNAs that were important or whether they actually had to be translated into protein. So to address this, we mutated the start codons so that we could make messenger RNAs, but they could no longer encode proteins. And what we found, so here are the wild type messenger RNAs that do encode proteins. We're expressing these genes in protoblasts. here are mutants where we've blocked the start codon so they can be translated. And what we found was that if we mutate the start codons, now these genes no longer have any effect on activation of autophagy. So you do actually require translation into protein for these to affect autophagy. So the question now is, what do these proteins actually do? And that we are still working on. So to summarize, we have ER stress, unfolded protein, activate IRE1. It has protein kinase and ribonuclease activity. Turns out BZIP60 splicing is not important for autophagy, and neither is protein phosphorylation by the protein kinase, but rather we've narrowed it down to this RID RNA degradation pathway as being important, and we found 3 genes that are degraded by the RID pathway and are negative regulators of autophagy. And now we're trying to figure out what these 3 genes do. And so to kind of summarize everything, we found that SNRK1, this protein kinase complex, regulates autophagy via the TOR pathway and also independently of TOR, depending on the stress condition. Auxin also regulates autophagy via the TOR complex. And then ER stress activates autophagy downstream of SNRK1 somehow, although we haven't made that connection yet, through IRE1B and via these 3 genes that we've just identified. And so overall, we now have some components to understand how autophagy is regulated, and we also still have a lot of questions for how these components fit together and what other proteins are involved in this pathway. And so finally, I'll acknowledge the people who have done this work. So Steve Howell and his lab are our collaborators for the ER stress work. They're experts in ER stress signaling, and we've been working pretty closely to try and understand what's going on in that pathway. And then June Marie is a graduate student currently in my lab who did all the SNRK1 work. And then a bunch of former lab members who worked on everything from the ER stress signaling to the TOR complex regulation work. And I will leave it there and be happy to answer any questions.
Could you flip back a few slides?
Maybe.
That's good. So when you did this RNA-seq experiment, now that you have these 3 genes, that was at the top 12. I mean, you probably have 1,000 genes to make the cutoff. So, well, the first question is, are you going deeper into that list? But the other question is, are there any other genes in the top 100 or something related to these lipid functions as well?
There are a lot of genes. So I say these are all ER-related genes, and right, and RID is a pathway that degrades ER-associated messenger RNAs. So your list is really biased towards things involved in the ER membrane already. So yes, there are lots more of these types of genes. I don't know how much you read into it, because those are the messenger RNAs. We're expecting to find messenger RNAs that enter the ER membrane or go into the ER and are somewhere within the endomembrane system or secreted. So we're kind of biasing ourselves already.
How much effort is it just to do those 12? I mean, like, how if you did another 12, would you get 3 more? Because obviously, it seems to me like you did a screen and you had a pretty efficient way of picking out the ones that were doing something.
Yeah, we can certainly go. We can go deeper into the list. It takes. We have to do them in transient. That's the problem. The problem and the advantage. So the transient assays are kind of time consuming, but it's certainly possible to go and look at some more. Yeah, we don't really know what to do with the ones we've got. That's one of the problems. Is trying. So we're trying to come up with a strategy. We're trying to come up with a strategy to how to really address these functions. knockout mutants don't really have much effect, but we're picturing it maybe having a more general effect, maybe on changes in the ER membrane and things like this.
So if you knock out, if you look at the Arabidopsis knockout collection, that doesn't help you?
Right, we probably would have to go and make double, triple knockouts and things like this. So we have looked in, we had a knockout, I think, in the B group 21. We know this is a glucosidase, and there are like hundreds of members in the family, and several of them are localized to these ER bodies. So there very likely is some redundancy in the function of those proteins anyway. And one thing that we're thinking about is whether these are really kind of direct regulators, or whether they're just disrupting the autophagy process and we need to get rid of them to allow autophagy to proceed.
So some RNAs are degraded more than others. That's how you found these by the rid.
Right, yeah, we just
RNA
looked at.
degradation. So is there some feature in common that ones that indicate that they have some kind of nuclease?
Yes, these stem loop things,
Yeah.
as with like everything to do with messenger RNAs,
Yeah.
it involves stem loops. And for the B60 splicing, there's this pair of stem loops that fits in. And it looks as though these messenger RNAs maybe have configured with a single site, and that they do have these stem loop structures that maybe signal them as rid substrates.
So it's like half splicing, it just
Yes,
does the cutting.
yeah. Mm-hmm.
And so it cuts at a single site, but then it gets degraded.
But then it's degraded. So it may be that under more severe stress conditions, it has relaxed specificity, something like that. And
Is that been published?
it's been suggested in yeast and things, but in plants, we actually don't. We don't know. We don't know. And the ER things are a little bit different in plants anyway, so you kind of hesitate to assume things from the yeast and mammalian literature.
And then, how do the mRNAs get to the ER? You said there's certain mRNAs.
Oh, so when it's during translation. So the proteins have the signal sequences exposed. So it's the mRNAs that are being translated.
So they're being translated.
At the ER.
Oh, okay. That's
yes.
important. Okay.
Yes. So it's the normal set of messenger RNAs that would be there, that would be associated.
Further question?
Does this RNA degradation have anything to do with ribosomal RNAs or ribosomal RNA proteins?
I mean, these are messenger RNAs that are being degraded, not ribosomal. Now, autophagy does degrade ribosomal RNAs, and that's a whole separate project.
Because I have a lead that's RNA that's I've been reminded of recently, and I just taking a leap here and wondering if it could relate to this at all.
Well, we have tentative data that the ribosomal proteins are reduced in translation in this.
Okay, so the RP46 mutant actually, every ribosomal protein is overexpressed.
Further question?
Yeah, I'm wondering if there is any tissue-specific response to stress
In terms of autophagy, we certainly see autophagy activated more strongly in some tissues than others. The problem that I have in drawing too many conclusions is that our assays work better in some tissues than others. So it's hard to do direct comparisons. But certainly, and when we do salt stress, the roots are being exposed more quickly and the roots seem to be activating autophagy more quickly. But the leaves will activate as well. I mean, it seems as though pretty much every cell type can actually activate autophagy.
Question? No, thank you.